Welcome everybody to today's Sverdrup lecture, visiting lecture series today. I just want to say personally, Dr. Dasgupta will give a very thorough introduction of our guest speaker today, but I wanted to extend a personal welcome and gratitude to Professor Ross for her time today, and I'm really looking forward to hearing your lecture. The Sverdrup lecture series is the final of our convocation series for the year and is sponsored by an endowment from the Sverdrup family and the Minnesota Space Grant. So thank you, Department of Physics, for that contribution. There are two goals of this visiting lecture program. The first is to provide an opportunity for our students, most of you out there today, to interact with, on a personal level, and I know you had a great lecture last night, with a scientist of national stature. So we're very fortunate to have Professor Ross here today. The second goal is for us of this annual forum is to have a wider scientific community. And so I know we have people here from all disciplines who have an opportunity to expand your knowledge and understanding of the work. And I'm particularly excited about this work that is on the boundary between two very important disciplines that fall in arts and sciences. So another great reason to be here today. Finally, this convocation kicks off a day of celebration for us, those of you that have spent a lot of time, students I'm thinking in particular of the last year, doing scholarship and creativity work, and we're going to have our Z poster session this afternoon, so I invite you all to be there as well, as you're able to celebrate all of that great work that many of you students in the room today have done. So I'm going to turn it over to Momita, Professor Dasgupta, to introduce our Sverdrup guest lecturer.
Good morning, everyone. It is my pleasure to introduce our speaker, Dr. Jenny Ross, today. Dr. Ross is an award-winning biophysicist interested in learning new physics principles in biological systems. She's working to better understand how living things organize themselves. She is motivated by the possibility of helping society by developing useful materials that can do things that biological systems are capable of, like self-healing. She won the Margaret Oakley Dayhoff Award from the Biophysical Society, the prestigious the Inspire Award from the National Science Foundation, and is a fellow of the American Physical Society. Dr. Ross received her PhD in physics from the University of California, Santa Barbara, and did her postdoctoral work at the University of Pennsylvania School of Medicine. As a Cottrell Scholar, Dr. Ross has pioneered innovative teaching techniques in active learning that are being adopted in many parts of the world today. Dr. Ross is also an advocate of women and underrepresented groups in physics, and she recently became the chair of the physics department at the Syracuse University. Let's welcome Dr. Ross today to deliver her talk titled "The Future is Alive: Physics Lessons from Biology."
All right, thank you so much. I think I'm going to move this over because I've got this one. Okay. This is really a fantastic honor. Thank you so much for inviting me. It was such a fun time last night interacting with the students. And I want to point out, I did go to a small school myself when I was an undergraduate. So coming back here and getting to see the science that the students are doing is one of the most exciting parts of the visit. I'm just surprised that, anyway, it's just a fantastic honor. I really appreciate it. I'm going to talk to you about some of kind of the big picture ideas of where I think the intersection of physics and biology can really go. Let me start by just having you imagine what the future could be if we really learned some of these lessons that we're going to talk about today. So just sit back and think for yourself, what would the world be like if infrastructure could do things like biology can do? So when you get a cut on your hands, your body heals itself. Wouldn't it be great if when a bridge starts to crumble, it could actually sense that it was falling apart and either redirect new material into the area or at least send out a signal so that we knew that it needed to be repaired before it fell apart. Same things for stuff such as, dams or oil pipelines, right? If they would be able to sense internally that there was something going on, we wouldn't have kind of oil spills that can contaminate our environment or floods that can cause when a dam breaks. And so, and in my personal opinion, what I really want to do is just make roads that can self-heal so I don't have so many darn potholes everywhere, especially at this time of the year before they've actually fixed them in the summertime. And so it would be fantastic if we could do something like this. We could create new materials that actually had the ability to heal themselves. Another thing that I want you to imagine is a world with personalized, instantaneous diagnoses and treatments. So some of you may recognize the picture on the far right. Does anybody recognize that one? Maybe some of the older generation. Fantastic Voyage. So you don't necessarily need to go watch this movie. It's not the best movie in the world. But the ideas of the movie are something that have driven many scientists, right? So science fiction is a place where we can be inspired about what we could accomplish in the future with science. And in this movie, what they had an idea of is what if you could shrink down to be inside of the human body? We are working on this now, right? Personalized medicine that has sensors that can detect when you have debilitating diseases, but just the very early stages. Even sensors like your Alexa in your home might be able to sense how you're walking and really start to look for early signs of diseases like ALS, which are adult onset diseases that you don't usually know about until much later in life, right? One of the first signs is that your gait changes. And so even just having kind of your normal room sensors that you have could start to understand and even diagnose your health. Ultimately, what would be great is to have something inside of your body that's able to report out to your doctor, telling you what's happening inside so that each person can get exactly the kind of medication or interventions that they need in order for our health to be improved. And then finally, one of my personal favorites, and especially now, is what would be great in the future is if we had clothing and fashion that could actually sense the environment and then change its properties so as to keep us safe. This is really important for our men and women in the armed services who are out in the battlefield and come across chemical warfare or come across biological warfare. If their armor would actually be able to sense and then change the porosity of their clothing so that they could counteract any kind of bad chemicals in the air so that they do not get hit with that and they can continue to do their work. But also in our current environment right now, right? If your clothing could actually sense when there was COVID in the air, wouldn't it be great if it could turn into a mask and then you wouldn't have to worry about, figuring out 4 days later that you had symptoms, right? So all of these things are things that we could do if we had materials that could sense the environment and then react to that environment. Sensing systems are not new. You have fire detectors, right? Smoke detectors. They turn on pipes, right? This is not something new, but this is something that makes our world better. It makes our lives easier to be able to do this. And I think what's different between sensors that we have now, where you need to report to a computer, right, and make an electronic analog that then responds out, is that we can actually create materials that they themselves are able to do this. And so biology has already done that, and so we can learn from biology. And so I'm just showing you now 3 movie examples. So on the first movie is an image of a set of cells that have been scraped across, and it's called a wound healing assay. So in order to study in the laboratory environment how cells are able to heal a wound, they actually induce a wound on a plate of cells, and then they can see the cells as they grow in together, right? So there's multiple things going on here. The individual cells themselves can move into the wounded area, but in addition, they can multiply purposefully in that area specifically in order to fill in the gap and create the monolayer of cells that they want to be, like you have on your skin. The middle movie is showing you a mimosa plant, which is one of these touch-me-not plants you may have seen. When they are attached mechanically that triggers an ion cascade down their branches, and it causes them to fold up. This is all a molecular level signal that comes from a macroscopic mechanical interaction, right? And then so it goes large scale, macroscopic, microscopic, down to ion channels, and then back up to macroscopic to cause the leaves to fold in. These are the same kinds of things that we could employ if we only knew how they worked. And then finally, a very classic movie of a cell going through cell division, which has the ability to completely rearrange its internal architecture, duplicate its chromosomal DNA, right, genetic material inside, and then divide it into 2 brand new cells. So each of these systems is showing you a different example of how biological systems are able to use noisy, kind of random, stochastic things, but then turn them into directed motion that is allowing real work. And when I say work, I mean from the physics point of view, force over a distance, right? Creating a force over a distance, work to be done. And that's where we get our energy. That's what energy is. And so biology inspires us to do all of these things. All right, I'm really inspired by sci-fi. I'm going to show a movie, and as soon as you know what the movie is, call it out. Ready? Do you know the movie? Which Terminator? Which one? Terminator 2, exactly. So this is the second one. This movie, although obviously an act of, artistic creativity, again, very inspiring to me because it is an example of what I call an autonomous material. A material that in and of itself can sense and react, and in this case, kill people. It's fine, it's fine. It's a robot from the future. That part hopefully is not real. Well, hopefully, I don't think time travel is possible. It's also not the smartest material. It does turn into a police officer. And then instead of just staying a goo and hiding under the person's bed, that would have been, this wouldn't have a movie if you just stayed a goo and then hide under their bed until they fell asleep, and then you killed John, whatever, it's fine. So the point is that it has the ability to do all of the things that I think an autonomous material of the future should do, like self-organize, move, sense, respond, right, to its environment. And so this is an example of what we could possibly do in the future, but not the killer robot part. No killer robots, okay. So I'm going to give you an example and lead you through how we can take biological things and be inspired about them. And we've done this many, many times already. So for instance, if you look at this picture of these horses, right? Horses are a biological machine, right? They are already out in the world. They've evolved after, evolution's, many billions of years of experiments and trial and error. We get horses, okay? What humans did when they looked at the horses, it said, that's interesting. I'm gonna harness that, okay? And so, so this is Stagecoach Mary here. She was a postal delivery person out in Montana, and she harnessed her horse in order to deliver the mail across the United States. She also happens to be a black woman, and I just found this so inspiring that she was one of the first people out in Montana actually delivering the mail, and she did that by harnessing horses so that they could go where they want to go. All right, so this is how we take biology and turn it into our own technology. Because after we did this, we thought, we can do that. We can do that better. All right, so this is Mr. Charles Patterson. He is one of the first automotive industry people. His car actually was bought by Ford and ultimately was one of the models for the Model A. And so he said, you know what, we don't need the horse. We can actually do this ourselves. And the nice thing about the car compared to the train needs the tracks, but the car doesn't, just like the horse doesn't need any tracks. And so we can synthesize it. So synthesizing it is when you take a biological thing and you turn it into a human-made thing. And then that, of course, can be taken and expanded upon, and we can innovate it. And now we have, crazy cars of the future, Teslas. We're going to have all kinds of electronic cars that are going to allow us to, do all kinds of stuff. And so this is an example of how we can take a biological system, be inspired by how it works, and then improve on it for ourselves to make it do what we want it to do. All right, so now we can look at smaller biological systems. So what I'm showing you here is a set of bacteria that are swarming, and you can see they're going all over the place, and they're not, and locally they kind of want to align because they're longer than they are wide, so like pencils in a box, they're going to want to go together. And now we can take this and we can do what we did with the horses. We can harness them. So what I'm showing you now here is a movie where a group in Europe has actually taken and created a very small rectifier that turns these bacteria, they swim into this like windmill. And so this windmill is being pushed around. It's a micro-scale windmill, and it's being pushed around by the bacteria. And the zoom out is showing you that you can multiplex this over many, many of these. And each one of these is now a bacterially powered energy source, right? Because anytime you start to turn something, now you've got a turbine. And once you have a turbine, you can create electricity, AC electricity on it. And so this is an example of how you can take a biological system that's already going to do its thing. You just have to feed it. You just feed it food, garbage, right? It eats its own self, right? And then you can turn it into something that you can harness. Once we harness it, now we want to synthesize it. So recent discoveries and recent work in the field of physics that's called active matter physics, the entire purpose is to understand active systems. Biology is inherently active. How do we know? How do I know that you are not in equilibrium, that you are at an energy scale that's above normal? How do I know that? I can stick a probe in you and I know it. What probe? Thermometer, exactly. So if I stick a thermometer in you, you're not at room temperature, right? That's because your body has a metabolism. That's the activity. So that is, so that's a question we don't really understand. This is what we're trying to understand. How do we use that activity in order to get things out? So one of the ways that we're doing this in physics is to make what we call Janus particles. So these particles are synthesized, human-made particles on one side. So they're called Janus because they're a two-faced particle. On one half of the face is a chemical reaction can occur. They usually put a metal like platinum, right? And then when they shine a light in the presence of hydrogen peroxide, it actually does a chemical reaction, which causes these to move. And so now we've synthesized an example that's just like the bacteria. And now we can get very cool phenomena out of just like we did with the bacteria, right? Okay, and then the next steps are to miniaturize it. So right now, these Janus particles are 10 to 1 micron in size. And ultimately, if we really want to create a material, we've got to start with constituents that are far smaller than that, right? So the things that make up a material, depending on what type of material you're talking about, graphene, for instance, is carbon atoms, right? Most of your semiconductor materials are like silica, things like this, right? So you need to be able to start with a smaller material. And so that's the goal of what we're trying to do in our lab, is we're trying to miniaturize it. So instead of having our power source being these relatively large bacteria that are 1 to 2 microns in size, we would like to have our energy source be even smaller than that, a much smaller little motor. And that is going to be an enzyme. So the enzyme that I'm showing you here is called urease. It takes urea in water and turns it into carbon dioxide and ammonia. And our first question that we had when we were starting to look at this is, can the urease enzyme itself run like a horse? Does it actually run faster than you would expect? Now, most things inside of a cell are diffusing, right? So I'm going to do a little dance. This is the diffusion dance, okay? So they just move around and they move randomly, kind of like these bacteria, but these bacteria will actually move forward before they rotate. But a very small object like an enzyme, it rotates very fast. So it's moving and rotating very quickly. What's powering that motion is not activity. It's just the thermal background. It's the fact that the water molecules are also all moving. They're hitting the object, and then they're moving around, okay? And so our first question was, when the enzyme is turning over, when it's doing its catalysis, when it's doing its chemical reaction, can it actually move faster, right? And what we found, surprisingly, is yes, it can. And that's really very surprising, okay? So the idea is that we have these little enzymes, and they take in a substrate, which we'll call fuel, and they expand out a product, which we call exhaust. And in this case, for this enzyme, the products are gases, ammonia and CO2. And so we know that we can power things using chemical reactions. That's how rockets work, right? In the case of most of our rockets, we have hydrogen and oxygen. We light a match underneath, and they form water vapor. So all the stuff you see coming out of those rockets is actually a water vapor, and that can propel the rocket up. The most important thing is that on the rocket is that you break the symmetry, right? So you don't want to have propulsion all around the rocket. You only want the propulsion at the bottom. So that's another thing we're going to need to do in order to harness the enzymes. We're going to have to break its symmetry. You can see right now it's got a threefold symmetry. So it's going to go this, right? It's actually shooting off in all the directions. So we need to break that symmetry. So the way that we're going to do that is, I'm going to show you that in a second. So this is just an example showing you that these individual enzymes do, in fact, have the ability to make themselves move faster. Okay, so what you're seeing on the left movie and the right movie are movies that are both playing back at 1 second per second. And on the left, there's no fuel. And on the right, there's fuel. And when they are diffusing, they're still moving randomly in what we call a random walk. But that random walk is faster on the right-hand side, and you can see it with your eyes. That's how fast it is. So the first time my student tried this experiment, she took the control data, no fuel, measured the diffusion, can see it, great. And then she put in the new sample with the fuel, added the fuel. And she came into my office, and she said, "Jenny, it disappeared." I was like, "What do you mean it disappeared?" She's like, "I can't see it anymore." I was like, "Well, did you put the protein in?" Like, you do like the basic stuff. Did you put the protein in? Okay, that's good. Okay, so that's good. And so we went in, and it turned out it was moving so fast that the frame rate she was using was not fast enough to catch the motion. She had to actually increase her frame rate in order to see the motion. And then you could see it with your eye, that it was physically moving faster. And so the whole point of science, right, is actually to try to disprove things. So in fact, when we were doing this experiment, I didn't think this was going to work. My thought was, some people have said this works, but we're gonna do it, and we're gonna show it doesn't work. When she came in and you could see it with your eye, then I was like, oh, well, I guess it's working. We better take a lot of data on this and do a lot of control experiments. I want to point out that the way that we're imaging this right now is with a type of microscopy called total internal reflection fluorescence microscopy. It allows us to see single molecules. So a single fluorophore is visible in here. We can see parts per billion in here. And so that is what's allowing us to see each one of this little dot, and each of these dots you're seeing is a single enzyme protein that has been fluorescently labeled that we're able to see with our microscope. Okay. And the thing, I put Einstein on here because the thing that's so crazy about this is that Einstein, one of his 3 big theories in 1905, one of them was diffusion, right? So what he did, this is a long-standing biophysics collaboration, actually. He took data from Brown, right, and from other biologists who had measured and looked at pollen grains diffusing in water. And he took that data and a whole bunch of data, and he said, you know what? I noticed that it depends on the temperature, it depends on how viscous the fluid is, and it depends on the size of the molecule. And he came up with an expression which said it's the temperature up top, and then it's divided by the size of the molecule and the viscosity. That is an expression that we use to this day. That expression cannot explain this. Nothing in that expression talks about activity. Everything in that expression is about thermal equilibrium, the fact that the water molecules are hitting you, and it's hard to move through the water because of your size and because of the viscosity. And so this is breaking down what Einstein said, right? How do we explain this increased diffusion motion of the enzyme? So we're going to need new theories in order to understand that. All right, so let's talk about harnessing it. So now that we've shown that the individual enzymes are like horses, they can move faster, now we need to harness it. So the way we're going to start doing that is using DNA origami technology. So we're going to make our nanoscale rockets out of DNA. You can take a big piece of DNA and a whole bunch of little pieces of DNA, and they will self-organize and fold into this scaffold. This is our chassis for our rocket ship. The nice thing about it is you can have little extra pieces of DNA hanging off. You can hook the DNA onto the enzymes, and then now you can decide where on the chassis you want it. Just like in the rocket ship, we need to break that symmetry. We need to make sure that we only have the enzyme on one side so that we can get it to blast off. And of course, it's not going to be a rocket ship, right? It's really going to be in our microscope, and we'll be able to see it. Again, with using that same technology of the single molecule imaging, we can also label the two ends of the rocket ship different colors. So I can see green on the far end and then red where the enzymes are. And not only will I be able to tell you which side is the front, which side is the back, but I'll also be able to tell you if it rotates and all other kind of information about it. And so what we're doing right now is designing new chassis for our rockets and trying to figure out what the best way will be to allow us to actually create an active system, like those bacteria, but now we're on the nanoscale. Right? So, and there's a big question here. Does the physics work at that scale? We don't actually know. But if it does, and then we can rectify it, we can harness it, and we can synthesize it, then we'll be able to start making those materials of the future. So that brings me to the end. I just want to point out, these are some of my students in my lab who are working on this work. This is from last summer. And I always have to thank them because they're really the people who do the work and allow me the time that I have to come up with crazy ideas for them to try in the lab. And I have quite a number of undergraduates here myself in this group. And so these are the people who are synthesizing it and the future of science, like all of you out there who are going to innovate it. And we're going to get where we need to be to make these materials of the future. So please remember to dream big and continue to be inspired. Thanks. Yeah. Okay, good.
Yeah, folks, please feel free to ask questions. Jade and Kong have the microphone, so you can raise your hand and they can come to you with the microphone.
Hi, I'm not great with microphone. At the beginning, you were talking about cells being able to replicate and heal themselves. But usually when the human cells, when they replicate, that's where you get tumors, cancer, and all that stuff from. If you're applying the same principle to a bridge that's trying to replicate itself, how do you control it so it doesn't over-heal itself or it doesn't over-multiply?
I think that's a fantastic question. I would love to be at the point where we need to worry about that. But no, that's a really fantastic question. I do not want concrete or pavement to actually decide for itself that it needs to replicate. So maybe, right, we actually need to have more control on it. I also think that this is a really important place as you build new technologies where we need ethicists, right? We need philosophers, we need social scientists to actually interact with scientists to try to figure out, like, where's the line that we need to draw on this kind of technology? So, maybe something for, instead of having your bridge actually heal itself, like, maybe what instead is it turns pink. And then you're like, oh, that bridge doesn't look good. That's a pink bridge. That's not good, right? And that's the signal to us that it's failing. And then you go in, either you fix it, or maybe, maybe it is driven by electricity. And you flip the switch on the bridge, it heals back until you see that it's not pink anymore, and then you put the switch back, right? I think there's a number of different ways we can still have human interfaces into these things as we need them. But you're absolutely right that you run the risk, right, of creating something that could be problematic. I told you I love sci-fi. There is a sci-fi book where people create a self-healing concrete and it takes over the world.
role model when you started to think about being a scientist?
Okay, it's kind of a cliche. I did read a biography of Marie Curie when I was in 9th grade. Actually, I was here. I was at Orono High School when I was in 9th grade here in Minnesota. And we had to read a biography, and that's what I chose her. You know, for me, it was really interesting because not only did she do this really amazing science and really dedicate her life to doing this, but she had kids. And she had family, and she, you know, made sure that she had time for all of those things. And I thought she was a really good role model for that. It was a different time, you know, kind of the way that people interacted. But she kind of showed me that it could be done. She actually did end up relying on her own father, who she moved from Poland to France to help her take care of her children. And so I thought that was very inspiring. I have a lot of like current role models who are kind of my friends who are in science now. And so, in fact, there's a woman who lives here, Tav Hawkins, who I was visiting recently, and she's also somebody I'm very inspired by, but didn't meet her until later in life. Yeah. Oh.
Hi, I'm a biologist that's physics adjacent, and I have a question about enzyme kinetics. So does this, you know, historically we've thought about enzyme kinetics as being diffusion limited, perhaps? Mm-hmm. So does your discovery, which I'm not aware of the details, make us rethink enzyme kinetics? That's part one. And a quick part two is, do you need to have your enzymes tethered to your chassis so they can move in 3 dimensions and maybe pull it? Yeah, so very inspiring talk, thank you.
Yeah, so as far as the enzyme kinetics, we have found that, in the paper that we published on this work, we do see that the activity, the increase in the diffusion, depends on the amount of substrate that we have there, so the amount of fuel that we provide. So we did do a scan of the fuel concentration, and then we show that it just looks like Michaelis-Menten, just like how you would expect. So is still that diffusion limit. One major question that we're probing right now with our new grant from the NSF is how much can these guys mix in the background? Because you're absolutely right. If you're able to then mix in the background locally, you will increase the diffusion of other items. Right? Other objects, the chromosomes inside of bacteria, for instance, or other things. And that's one of the first indications that they had that enzyme activity could act as what we call an active bath. So as opposed to a thermal bath, where you're just being driven by temperature, an active bath is where you're driven by the random activity of the objects inside of the bath. And so in that case, if you are able to basically stir, you would be able to increase your enzymatic activity. The other thing too is all enzymes obviously are temperature dependent, and so that's another, there's an idea of an effective temperature that you get due to stirring, but then there's actually a real temperature, and many enzymes are exothermic. In fact, the one that I look at is exothermic. We have no indication that in our experiments where we're really very dilute, right, 90 picomolar is what you're looking at when you see single molecules here. So they're not capable of really creating a furnace in there that will heat everything up. If you have enough of them, then you will actually also locally heat and change the viscosity. And all of those things will add to also enhancing the activity. What was your second question? Oh yes, so the system I'm showing here, I didn't explicitly draw it, but they are tethered. And you're absolutely right, I'm showing this as if these enzymes would be what we call a pusher in active matter, but they could be a pooler, right? And so, and in fact, you should actually look at some of the literature about the Janus particles. There is a group that's been using this exact enzyme to coat large objects. So these are nanoscale objects, but he's been coating large beads or large pieces of like glass with these enzymes. And then you can see that the enzymes are actually able to propel it. Now with those, because you can decorate the surface and you know which side has the enzymes, you can actually tell if the enzymes are leading, the Janus face with the enzymes are leading, or if they're trailing. And that's the difference between a pusher and a puller. There are Janus particles that are pullers, where you literally change basically the viscosity or the osmolytes in the front, and that actually is what drives the propulsion.
You talked about how like it's similar to how when we get a cut, it would heal. But like we have things like if bacteria gets in the cut while it's trying to heal, it has issues with that. Are you having issues with like outside environmental things getting inside these things? Are you worried about that for the future?
I think you would have to worry about that just like you'd have to worry about like concrete tumors, right? It's kind of a similar thing. You would expect that ultimately the environment might evolve to affect your, or not even evolve, just somehow there's a good niche for a certain bacteria or a certain thing. We don't have to worry about that because I'm in a very sterile laboratory environment, but ultimately if you were starting to deploy these as infrastructure, you would want to, you'd have to test it in an outdoor environment and make sure that that all continued to work, and it might not. I think there's many, many places where this can go, right? And so I think there's just, we have to try, and then, but try it in a, ethically responsible way, right?
So I'm a mathematician, and I'm just curious, like with your research, have you found any new sort of areas and discoveries that have maybe led to more interesting mathematics that you weren't expecting at the time?
Interesting. I'm not sure if there's been any new work from my work on mathematics. There are other people who study kind of soft matter, like soft materials, and there's some really interesting things going on, in the liquid crystal field, where it really does bring in the kind of geometry in with the structures that liquid crystals can form. There's also people who work on, or like not DNA origami, but like actual origami, and how you can get it to fold and actually creating, materials that self-fold. And so that actually would be a very interesting platform with this, right? Where you can have the kind of like a thin sheet, and decorate it on the surface. Maybe you have some areas that are sensors and some areas that are kind of actors. And then when they sense and act, then you could have it fold up and then, or redeploy into some other activity. That could be also kind of a base subunit. We chose to do the DNA origami here, but one of the things we had considered was actually a thin material that would be deformable. That work has actually a lot more connections with the mathematics because of the thin sheet mechanics. There's a lot more interesting things going on there as far as the theoretical and mathematical side.
So I understand that you're like harnessing the energy from the enzymes to move the like nanoscale rocket, but what are you trying to like get the rockets to like do, or what do you like see that for? Like could they be incorporated in like the, like the diagnosis of stuff like being, or just like what's your like goal for the rockets basically?
So once you have an active particle, you can rectify it, right? So what you saw with the bacteria where they could then push the kind of windmill around, that's a type of what we call rectification, or like making a diode, right? Like they basically can go one way but not the other. And then by using that, you can get it to do work, right? And so that's one thing. Another idea is, again, just using the randomness, like not even worrying about the directionality of it, but to just use that to fluidize or create an active background bath in your material, right? And so that you would be able to then do other things at room temperature that normally would only occur at a higher temperature, because this overall activity gives you an effective temperature. And right now, we're right at the beginnings of this, but we do think that enzymes are able to do this inside of live cells now. If you shut down all the enzymes in a live cell, the whole inside of the cell gums up and basically becomes like a glass. And so in order to kind of, the enzymes themselves seem to be fluidizing everything. We'd like to know if these, which are kind of in the in-between scale, but also are programmable, would be able to do that. And then I'm only showing you with the enzymes at one place, but we might be able to, we can actually decorate these with the enzymes anywhere along the length. So you might imagine that we decorate it in a spiral, and then we actually make spinners that rotate. And then if we did that, that would then create a different form of kind of activity inside the bath that might move things in a different direction or fluidize in a very different way. We're really at the fundamental, basic science part of this kind of set of experiments. And so I'm excited about it because I think there's a lot of different directions that this can go. Yeah.
I was just curious with your DNA rocket here. Like, I see 6 cylinders. Are each of those like DNA double helices? And why are there 6 of them? And how did you decorate only one side of it?
Right. So this is called a 6-helix bundle, and it is actually 6 double helices. So they're about 6. The whole thing is about. In fact, and this one says 16 nanometers, but the one that we've created is actually even thinner. It's about 6 nanometers, and so it's one giant round piece of DNA that comes from a bacterium, and then you put that in with a bunch of things, little pieces of DNA that we call staples, and they have connections, but like across the circular DNA, so that it causes it to fold up. So the circular DNA obviously doesn't go around as a circle. It goes around as like a bunchy ball. And then as those staples come, when you lower the temperature and it starts to bind, those staples come and they can actually cross-link between different parts. And that's how these DNA origamis pretty much all work nowadays. And so those are double helices, but they're not necessarily all one piece. They can be made from these staples that are cross-linking them into that double helix. The staples can also have additional DNA that hangs off. And so it's that additional DNA that we then have a partner DNA that's bound to the enzyme off of its N-terminus, right? So just using kind of basic like chemistry that you can buy from Fisher, and you can click the DNA onto the enzyme, and now you've created a base pairing with the little hairs that are hanging off, the little DNA hairs that are hanging off. So we can control when we change the structure which staples have an extra little bit hanging off of them that allow us to bind the DNA. And as long as the little pieces of DNA are far enough apart, because we end up getting exclusion, like a parking problem, where like if one comes here, then those enzymes are actually quite large, and the next one can't really bind right next to it. So that's a lot of what we're doing right now in the programming of this, is trying to figure out like how close can we get those pieces of DNA, how many of those enzymes can we get on the end, and then how close can we get those together. Yeah.
That's really cool. Could I ask one follow-up question to the fluidity thing? Like, I'm wondering if this is like a biological, like an evolutionary process. Like, have you considered heat shock and cold shock responses? Like, maybe enzymes are helping fluidity when a cell is undergoing temperature stress.
Yeah, maybe. To be honest, I haven't taken biology since 10th grade, so I know about the biology that I do, and I do think that we know that there are processes that turn on and off, right? When you have heat shock, or when you do other kind of disruptions to cells, right? I think that these enzymes, like I said, I think that making a protein in the cell is hard, right? I think that making, folding it and getting it to be correct is hard. And so biology then has double or triple duty for almost all proteins. And so I think if there's an additional activity that just, any enzyme in the cell, it's doing its chemical reaction, maybe it's inside of a membrane, inside of an organelle, maybe it's outside. It has its first primary function, but there's no reason why this couldn't be a secondary function of all enzymes, to just fluidize the inside. And I also have a personal feeling that if it helps biology, if it survives, then biology can do it. So there's a really cool field right now in biology, or in physics, called quantum biology, when they're starting to see quantum effects in biological systems. And I'm not working on it, but I'm really fascinated by it, because It's this. I think if a biological system had to add 1 + 1 to get 2 to survive, it would just do it. And so if a biological system needs to have quantum mechanics and have quantum coherence in its structures in order to do whatever it needs to do to survive, it's going to do it. And so it's a really cool subfield right now of physics, and there's been lots of talks about it at March meeting, the big physics conference. So.
Hello. I had a question about, with urease, the force that's kind of increasing this diffusion or activity. Is that like, do we know if it's the release of the product or like a conformational change that happens in the reaction? What is actually making it go faster?
That is an awesome question. If you're interested in come and work in my lab and try to figure it out. I don't know the answer. Nobody knows the answer. So multiple labs now. When I first started this project, people had seen this, but they'd only used one technique ever. And we know in science that that's not the best way to do science. You need to approach the same question, especially one that's weird, like this one, and somewhat controversial, with multiple directions. And so we were the first ones to employ this single molecule direct imaging, but people had previously used a correlation technique and shown that it does it. Now people have used scattering, they've used a number of different techniques in order to show that indeed it does look like urease is able to do this. And in fact, there was one really cool technique where they used a laser spot, they focused it down, and instead of looking at correlations as the object went through, as the enzyme went through, they just looked how long does it take for it to pass through the spot, right? And so of course it doesn't always go through the middle of the spot, sometimes it goes to the edge, and so you get a distribution of kind of time of flight through the spot, right? Then what they did is they used a second laser around it, because there's a minimum that you can do with diffraction. They used a second laser around it to actually make that internal area smaller. So that external donut laser actually turned off the fluorescence. So they made it smaller and smaller and smaller. And when they made it small enough, they actually saw two subpopulations. One that was moving way faster than it should have, and one that was moving at what you would expect. And the ones that was moving way faster were the ones that seemed to be doing this kind of enhanced diffusion through that spot. That, I thought, was a very nice set of experiments, right? Very direct. You're just like, how long does it take for it to pass through, this area? And in that, they saw that this enzyme was able to pass through faster when they got the spot to be around 50 nanometers. So over a 50 nanometer, and they can measure the time it takes for it to go 50 nanometers. Now you've got a velocity, and it's moving really fast, like very surprisingly fast. And so I think the mechanism is still a big question mark. We do not know how it's happening. It's going to take more experiments of a variety of kinds to figure it out.
Thank you.
There's a couple of hands back here too.
So obviously you're in the beginning stages of it, but when it does get to the real world, what would be like your ideal first place that it goes, or like bridges is like a really big thing, but what would be the small thing before that where you kind of see it in the real world? I really want to cure the world of potholes. And maybe this is because I live in Syracuse, but I think that is a thing that you can do stepwise, right? So people study pavements, people study cement, and you would basically, you dig out and you make a cement, and then you patch, and then you can do experiments in outdoors, just in small patches where you're inducing cracks, you're inducing potholes, and then looking for the self-healing. People are working on this in other directions as well already. So there are people who have tried to create vasculature. So the way that your blood goes through, because you have veins, right? So the idea, could we put veins and arteries into the concrete to deliver the material? And one of our problems right now is very different from the way biology designs, is that biology is water-based, right? And we don't have water-based pavements, right? But how do we deliver the new material there and maybe get rid of the old material? And so that's one direction that people are moving And they're learning from biology to do it, right? Vasculature is something that we have in our bodies, and they're now trying to add it to the concrete to give you kind of the ability to have more material show up where you're losing it into the cracks or the potholes. So I do think that that's something we could do. My method is very different because I really want the whole material to be, machines all the way down, right? And so the idea would be that, you think of this material as this inert thing, but no, it would actually itself be a machine. It would be a material machine, right? And so, but there's lots of different ways that we can approach this. Yeah.
I know you talked about the thermodependency of the rocket and the enzymes, but because they're enzymes, I was just wondering how dependent they are on the pH of the environment that they're in, and the function, and how the pH affects the function of the rocket. Like, does the function stay the same or worse or get better in different pH environments, and how does that affect the application beyond like a lab?
Yeah, I think that that's a really great question. In fact, our enzyme that we use, one of the issues with it is that it can increase the pH as it functions. So you're releasing CO2 and ammonia into the liquid, it can increase the pH. Of course, everything we do is in a buffer. The whole point of a buffer is to maintain your pH at a set level. So it changes slowly, especially when we use a very low concentration like this. But I absolutely agree that in addition to changing the temperature possibly, or the actual chemical environment, changing the pH is something that can affect it. We are currently running experiments at pH 10 to see how much the pH just affects, everything as a kind of control experiment. We know, you're right, we know that enzymes actually do respond to pH. One thing that would be really cool with this, again, using enzymes, is to create enzyme, pairs or, cascades that can occur where you're actually you'd use a second enzyme to clean up the pH and then bring you back to the substrate of the first reaction that you want, right? That would actually only help my idea, right? Because every enzyme in it should also be contributing to that active bath, that fluidization, right? With the way I'm thinking about it, every enzyme would be adding to that enhanced activity and that enhanced thing. So that's one of the things that we are trying to do. We also have a microfluidic device where we have semi-permeable walls in our microfluidic device. And so we're able to continuously feed our enzymes as well as continuously remove product. And so there's the cascade kind of reaction cycle idea, but then there's also just like brute force. Like, can we just remove product and continue to provide fuel to the enzymes while we do the experiment? And so we actually have that microfluidic device kind of up and running, and we're starting our first experiments with the enzymes in it now. Yeah.
I wonder if anybody in your lab has thought about applying this to plastics, because plastics can be a little bit difficult to reuse and recycle,
Right.
Instead of just having like concrete take over where it's more
Yeah.
permanent, and it can be a little bit more difficult to ensure a larger structure if you guys just replaced some component of plastics instead.
Yeah, there are people working on like polymer systems that are self-healing, like these vitrimers, where the idea is that they are actually polymerizing when you break them. They can repolymerize kind of on the fly. But yeah, no, that's a great idea to think about, plastics, and then also, like you said, recycling of the plastics. It'd be fantastic.
So I guess just thinking about like the microfluidic device where you're able to remove product and add substrate whenever you want, or in theory whenever you want to, how would you apply that to like potholes, for example? Because like they're just like, it's a road, right? How would you apply adding more substrate or removing whatever product you want to get rid of out of your pothole?
Right. So I think in some sense, the microfluidics that we're designing are similar to like the idea of the vasculature, right? So we are kind of pumping in and pumping out. And so having that kind of a network of pipes, and maybe they're flexible, maybe they're made out of also a material that's semi-porous, that's allowing, things through. Or maybe there's a switch, you turn it on. There's like at night, there's a road crew that comes around and like, yep, pothole, turn it on. Flow, 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 refill the pothole. And then when the potholes are filled, turn it back off, right? So all of these things, like ultimately, I would love to see a world where we really had autonomous materials, where you didn't need the human intervention, but intermediate points could include the human intervention. And so, I think that that kind of having that vasculature so that you're giving it more material, and then you're actually able to stop giving it the material too, helping with the tumors, as we talked about earlier. Well, thank you. Oh, thanks.
Was a great talk. I would like to now welcome our student, Grace Puchesela, to give the vote of thanks. And folks, we have some light refreshment just outside, so please feel free to help yourself before you leave for your classes. And also this afternoon, we have the Z, which is the annual celebration, and students would be presenting their posters. This is first floor Hackfers, so please plan to be there if you're already not presenting, to encourage other folks who would be presenting there. Grace.
Hello, everyone. Thank you for coming today. My name is Grace Puchesela, and I am a senior student majoring in biochemistry here at Augsburg. Today, I would like to thank Dr. Ross for, on behalf of Augsburg community, for coming to visit us and be able to share about your scientific research and how it's relevant to our shared future. We will also like to honor your leadership in supporting inclusive environments within the STEM field. And to express our gratitude, we would like to give you this gift, which includes, so this gift, which includes your very own Augsburg water bottle and stickers to celebrate your talk in the Sci-Sagathon, which is an event to celebrate student research and scholarship later today. We are so excited that you will be joining us and be able to interact with some research students and faculty mentors. Thank you.